I'm Pin Chao. I'm in the Institute of Molecular Medicine and Department of Life Science. So I know some of them may took my advanced cell biology class, and some of them molecular biology advanced class. So today I will introduce the automatic tracking program of mitochondria, and especially designed for live imaging. So in today's lecture, I will give you a brief introduction of mitochondria dynamics. And then I will show you some of methods previously developed for the mitochondria analysis. Then in the final part, I will introduce this paper. All right. So I think that all of you may know mitochondria are one of the most essential organelles in cells. It contains double membrane structure, outer membrane and inner membrane. Between two membranes, there's the intermembrane space. In inner membrane, there's matrix. So like TCA cycle enzyme located to matrix, and electron transport chain located in the inner membrane. The most well-known function of mitochondria, probably everyone knows, is ATP production. However, in addition to ATP production, it has several functions such as calcium homeostasis, redox homeostasis. They also produce a lot of metabolites for essential metabolites. In addition, mitochondria are the major source of reactive oxygen species. So under the basal conditions, reactive oxygen species are the important signal molecules. However, if there are too many reactive oxygen species, they can induce oxidative stress. So mitochondria are not stay in cells. They are dynamics. So they include the mitochondrial fission-fusion process, and they can move. Okay, so you can see these are all mitochondria. Some are red, some are green. So you can see the green mitochondria can fuse with the red, and then they can do the fission process, like here. They can fission and the fusion. And they can move. So why do they need to, why are they so dynamic? Because for fusion, they can kind of dilute the damaged materials in mitochondria. For example, if there are too many damaged mitochondrial DNA, through the fusion, they can dilute the amount of mitochondrial DNA. And then during fission, if mitochondria or cells are under stress, they can kind of put back bad material to one side, and they can do the fission. So they can put the damaged stuff in one side and keep the healthy in the other side, and then during the fission, so the damaged mitochondria can be engulfed and degraded by mitophagy, so mitochondrial autophagy, and then so keep the mitochondrial quality. And through the fusion, like I said, they can dilute damaged stuff and then or increase the energy use. And then, so in the mitochondrial fusion, there are two major kinds of proteins. One is mitofusion, and the other is OPA1. So they are all the GTPases, and mitofusion located to mitochondrial outer membrane, and the OPA1 located to mitochondrial inner membrane. So during fusion process, mitofusion and 1 and 2 can connect together and help the mitochondrial fusion. So in mitochondrial fission, it's involved in several different kinds of organelles, including ER and actin, cytoskeleton. So when mitochondria need to undergo fission, actually mitochondrial DNA will come too close to the fission site, and then ER will mark the site of fission. So after ER comes in, and then they will recruit the actin filament. So the ER mitochondria can form the actin filament for the first round of contraction. And then they recruit the DRP1, which is the most well-known fission protein. It's also the GTPase dynein-related protein 1. And then they can, after recruit the DRP1, they can wrap the mitochondria and then do the fission process. So this is the very basic idea of mitochondrial fission and fusion. And for mitochondrial transport, so I will use the specialized cell type, neuron, because neuron is very asymmetric. You can see the cell body in one side and a very long axon and the synapse in the other side. So from 1950s, it's already known mitochondria can locate to synapse area. So this is the image, and then here the neuron, and then here the synapse. So you can see the small vesicles is the synaptic vesicles in synapse, and then here are some mitochondria in the ER. So we know that synapse need to release and recycle synaptic vesicles, so they require a lot of energy, and they also require the calcium homeostasis. So therefore, because mitochondria, most of mitochondria are synthesized in cell body, and so the mitochondria need to transport a long way from the cell body to synapse to provide the energy and calcium homeostasis. So some dysfunctional mitochondria can go back to cell body for recycle. So this is the Drosophila neurons inside larva. So the larva of fly, so we can dissect fly larva, and here the ventral ganglion, which cell body is located here, and you can see very long axons. So it's a segmental nerves, and then they can attach different segments of larva. So the green is mitochondria tagged with GFP. So you can see in these movies, you can see some mitochondria move from cell body to synapse, some move back, and some are stationary. So people think that because the cell body just generated more energized, more functional mitochondria, and they need to transport to location where needed. So this one is called from cell body to synapse, we call the anterograde transport. From synapse back to cell body, we call retrograde transport. And it's probably because these mitochondria are not that functional, they need to be degraded, so they can be transported back to the cell body and degraded by mitophagy. And you can see some stationary mitochondria, probably, I think that they are located there because they need that. So energy, they need a lot of energy in several areas here. So during the axonal transport of mitochondria, if it's the anterograde transporter, they use a kinesin as their motor protein, and the retrograde, they use dynein as their motor protein. And between mitochondria and motor proteins, there are some adapter proteins. So how do they know mitochondria need to stop there? It's because of calcium. So when calcium levels increase, they can bind to the mitochondrial protein mirror. It's a calcium sensor. After binding to mirrors, the whole motor protein with mitochondria may leave the microtubule, or the mitochondria itself leave the motor protein. So basically, if the calcium levels increase, the mitochondrial movement will stop. Okay, so this is the very basic idea of the mitochondrial dynamics, and it's already known that if mitochondrial dynamics are dysregulated, they will induce several diseases, especially in neurons. Several neurodegenerative diseases has been shown related to mitochondrial dynamics, such as Alzheimer's disease, Parkinson's, and Huntington's. So therefore, it's important to study the mitochondrial dynamics in different model systems. So that's why several groups study the mitochondrial dynamics. And at the very beginning in this field, people use manual tracking. So basically, they check by eyes. And this is very time-consuming and labor-intensive. So in my PhD, I studied the axonal transport of mitochondria. At that time, I still use manual tracking. So basically, we took the images, then we use images and click forever and get the data. So now there are more groups try to develop the automatic way to analyze the mitochondrial morphology and the transport. So I will introduce some of them, not every algorithm, but some of them I have used or some famous one. So the first one called mitograph. So this is original design for yeast mitochondria. So you can see this is the yeast, budding yeast, I mean, and this is the mother cell, this is daughter cell. You can see the mitochondria are most like tubular structure in budding yeast. So at the very beginning, mitograph designed for mitochondria, yeast mitochondrial morphology, and especially it's great for 3D images. So all the algorithms are good for 2D images, but not many of them are great for 3D images. So this one, the original design for 3D imagery and the yeast mitochondria, so they are more optimized for tubular mitochondrial network. So they have two versions, and in the version 1, you can just, after you acquire your images, and you can crop them and then send to the website. So they will do everything for you. And then you can get two kinds of data. The first one is the surface volume, which means the voxel occupied, the volume occupied voxel. And then you can get the skeleton volume and the node. So the node, which means that if there are branches in mitochondrial network, they will give you the branch size is one node. So from this one, you can get the basic morphology parameters of mitochondria, including the length, surface area, volume, and then the good thing in this one is they give you whether your mitochondria is more like a network one or more like just tubular one. So if there are more nodes, which means there are the mitochondria are more like a network morphology. Okay, so I have to mention that, so they developed version 1, now they develop version 2, but there are no website anymore. So they can, you can download it. I think it's written by Python, but it's incorporated in ImageJ. However, they only write the Mac version. So for now, if you are a Windows user, you cannot use this algorithm. And then the version 2 that applied the mitograph to mammalian mitochondria, it seems that it works pretty well. You can still see the tubular mitochondria, and then there should be some knots in here. So anyway, for the mitograph, it's great for the mitochondrial morphology analysis, 2D and 3D, and especially in yeast, but the mammalian cells now I think it's fine. But then one drawback is they don't have temporal function. So you cannot see, if you took the movies, you cannot use this one. Okay, the second one is the QVP, which is developed by Thomas Squaw's lab. So in this one, this algorithm is special for tracking mitochondria, not for morphology. And then this is also is an ImageJ macro. So the idea is that if you put the cytosolic GFP, you can use the GFP signal to draw the cell outline automatically. And then in mitochondria, they automatic segment mitochondria and give them the mitochondria. So how do they know mitochondrial movement? They quantify the variance of pixels. So if this is the first frame and the second frame, and if you see the pixel intensity difference, they will call this its movement. So you took a movie, you have several frames, so they incorporate everything and then calculate the variance of the pixel. So according to this number, they will say, okay, so these mitochondria are more motile or they are more stable. So the good thing is it's very straightforward, and then you can, they will show you the heat map. So visually you can see whether in these cells, these mitochondria are more motile or more stationary. But they didn't give you the parameter of individual mitochondria. So you cannot, from this organism, you can only know, okay, in this kind of cells, mitochondria move probably faster. But you cannot know, okay, how about this individual mitochondria? You don't know. They only show you the heat map, and you can know, okay, that is, okay, probably in this kind of cell, mitochondria move fast. That's it. And they don't give you the morphology parameter. They only show you the heat map. Okay, so this is the QVP, and the third one is TrackMate. So TrackMate is also good for 3D images tracking, and then you can see they can track the individual object. However, this one, the first design is for C. elegans cell development. You know that C. elegans development is from one cell and then divide to two, and then they can track them, use this TrackMate. So because at that time, the C. elegans cells are more round, so it's specially suited for the round spots or particles. And then, but it's good. You can track the single object. But because in mitochondria, they are more like tubular form, so it's not that good for the mitochondria tracking. However, very recently, they developed a new version called TrackMate 7. And in this one, there are, I think it's a lot of progress. The first one is they incorporate several different automatic segmental tools. So which means that they can automatically let you know this one is one cell, this one is another cell, so they will segment by themselves. And because they incorporate different kinds of segmental components, so the function is improved a lot. And then now they can track the not only the round object. If you see like this is the moving cells. And they can also check the object in different kind of shape. But because it's pretty new, they didn't apply to the mitochondria, so we still don't know whether it's good for mitochondria analysis. Okay, so for the axonal transport, there are not that many algorithms to automatically tracking. So in 2013, so this is from my PhD group, we still use manual tracking. So you can see this is the axon and the green one is mitochondria. So you just check frame by frame. So like the red arrow here is the anterograde. The blue arrow is retrotranscript. Okay. So we can also photo bleach the segmental nerves, and so it's easier to see the mitochondria move into this area. So now, if you want to automatically track the mitochondrial transport in axons, the most convenient way is you generate a chemograph. And then you can analyze different parameters by chemograph. So this is chemograph. So the x-axis is the length of the exon from beginning to the end. And the y-axis is time. So if you see this line here, from here to here, which means that at the time 0, this is located here, and by time pass, they move from the very beginning to this area. So this, if the like this, is called the anterograde transport. If you see the line like this, it's retrograde transport. So if it's a vertical line, which means after time passes, the mitochondria locate in the same spot. So this is stationary mitochondria. So by analyzing chemograph, you can get many parameters of mitochondria movement. For example, you can know how many percentage of mitochondria move anterogradely or retrogradely or stationary. And you can know the speed of mitochondria. You can calculate the slope of the line, and then you can know the speed of mitochondria. So this is also from Thomas Squat's lab, and called the chemolyzer. You can use this one to analyze the mitochondrial movement. So the idea is pretty simple. So first, they will automatically generate a chemograph. So you can use if you have the GFP signal in your exon, they will automatically recognize this exons and then draw the track of the mitochondria movement. Then they will automatically generate the chemograph. However, this one is like a semi-automatic way, because after generate the chemograph, the program doesn't know which line is one mitochondria. For example, it's possible that this one is stationary mitochondria. Somehow here it starts moving, but then in reality, probably this is all the stationary mitochondria. So you have to assign at least some part of them. So you can assign this one. You already know this mitochondria moves like this, so you will assign this is a mitochondria. Then after you assign some of them, they will automatically recognize the rest of them. And then they can use the chemograph to analyze the parameter. So as you can see here, the vertical line here shows the stationary mitochondria, and the motile, the line like this, is a retrograde movement. And the line, this is anterograde movement. So you can calculate this by chemolyzer and then know the movement parameters. Okay, so far, any questions? No? Okay, so now we'll move to this paper. So this group, Dickman group, developed automatic segmentation and tracking algorithm called mitometer. So it's not suited for exonal transport. It's only for the more symmetric cells. Like probably most of you use like hex cells like this. They develop this algorithm for that. So I mentioned, so in the previous method, although there are some good advantages, but there are some drawbacks. Like mitograph, it's not for temporal, temporal movies, and then for QVP, they didn't give you the individual information of moving mitochondria. And like TrackMet, it's only for the wrong object. So in this group, they developed this new method, so you can automatically check the mitochondria. So they also improved their segmentation method, and then so you can just input your 3D or 2D images, the time frames, and then the first step, they will do the background subtraction. So they will use the diffuse background and then subtract the background. So you know when you take the image, some mitochondria or some areas the signal are stronger, some are lower. So by diffuse background subtraction, they kind of minimize the difference between the very bright area and the very dim area. So that they let the signal to background ratio like more similar. So after they subtract the background, so this is the original image, and this is after the diffuse background subtraction. So you can see after this subtraction, here the original here the mitochondria are very bright. Now it's not that bright, but they also remove the background signal. The original area is pretty thin, but they still keep the try to keep the similar signal to background ratio. So that these two areas, this is very bright, this is very thin area, that the signal to background ratio become similar. Then they can do the threshold and then do the mask of mitochondria. So do you know the mask in imaging processing? What do you know what that means? No. Okay. So so when you take an image in mitochondria, you know the intensity of mitochondria, right? But for mask, which means that if there is mitochondria signal there, so I will give you, okay, this is 1. So you can assign, okay, the mitochondria here is 1, others are 0, they are all background. So from this, that you don't know the mitochondria intensity, you only know the location of mitochondria. So why do we need this? Because after you get the mask of segmental mitochondria, you will know, okay, so in here, I don't care about the intensity, I know there is mitochondria. So you can apply this mask, go back to the original mitochondria. So if there are some blur area, something like here, after you give the original images with mask, you can remove this like blur area. So you can assign, okay, so I just want to focus, analyze the intensity in real mitochondria. Don't give me the number of the like diffuse signal. So after that, they apply the mask back to the original images. So and then they can do the pretty good segmental. So this is the original image. So this is a time lapse, as you can see. And this is the diffuse background. After subtract the diffuse background, you can see the signal to background ratio is pretty similar, and then it's easier for thresholding. And after thresholding and give them the mask, then you apply it back to the original image. So this is the final image you can see. So I have to say, after you do that, apply back, you still keep the intensity in this one. And then you can calculate. This is just assign the mitochondrial location to the original image. Okay, so then after that, you can use this segmented images and then do the following analysis. Okay, so now after automatic segmentation, now then start to try to track mitochondria. So it's hard for algorithm to know this is the mitochondria I need to track. So, and then how do they know after different time frames this is the same mitochondria or this is different mitochondria? Are they in the same track or they are different track? So they apply this method, try to differentiate the same track or different track and the same mitochondria. So at first they assign each mitochondria a track, which means they will track them. And then they will collect different parameters in the mitochondria, including the distance in between, and the axis, the z-stack, the z-axis, intensity, volume, and the solidity. So do you know the solidity? So this is another morphology parameter to show how compact your object. So basically, it's your object volume divided by the volume like surrounded to the your object. So if your solubility is higher, so which means your object is more compact. So they collect different information, including distance, the length, volume, then they compare each frame. So if you compare the second frame to the first frame, and you get a high correspondence in these parameters, which means that they are the second one compared to the first one. All the parameters are pretty similar, which means that they are the same mitochondria. So the cost of assignment is lower. So the cost of assignment means that they will know whether these are the same mitochondria. And then if these two mitochondria, they have low correspondence or the high variance in these parameters, which means they're probably not the same mitochondria, and then so they will give you the high cost of assignment. So after collect this assignment cost, so how do they know it's the confidence of assignment? So they will run the algorithm and then try to find the lowest cost of assignment. So they will say, this is confident assignment. So far this one is okay. So which means if there are two frames, the parameters are similar, they are okay. So this is low cost. If they are very different, it's high cost, and they will try to lower the cost of assignment. And then they can start to track. If they track the mitochondria and then know the entire tracking of the mitochondria, all of them are confident assignment, they will say this is a confident track. So the tracking, when they track them and the confident assignment, they are there is confident track. But sometimes they also want to know to try to further confirm whether this is the real track, and then they will compare the coefficient of variance in each parameter in the track or between different tracks. So they will try to differentiate whether the old track here is indeed the real track or it is combined with different kind of tracks. And they also can differentiate the track 1 and the track 2 are different. So they also use the variance or the parameters in the previous slide and then give them the weighting. If the parameters between two different tracks, they will give you a higher weighting. But if the parameters are different, very great in the same track, they'll give the lower weighting. And after compare this, they can, after several rounds of the tracking process, they will know, this track is confident, and that you can differentiate of different track. So, and the final one possibility is that when you take the image, it's not always perfect. Probably sometimes in some images, your mitochondria disappear. It's not really disappear, it's probably just covered by other signals. So in that images, you didn't find the mitochondria that you are tracking. But in next frames, the mitochondria pops out again because it's leave the covering signal. And so now you have many different choices. So how do this algorithm know which one is the real one? So they can combine the track in the after disappearing and before disappearing, so they can combine together as a track. So they give some parameters like if the degree, the travel angle below 20%, they will say, okay, this is probably the same. And the distance lower the max velocity, they will say, okay, this is probably. So combine these two criteria, they can combine the track together. And if there's only one possibility, they will combine as a track. So they will do this several rounds to make sure this is really the track without, like this is not the new track, this is the same track. Okay, so far they use 3 different methods, try to automatically track the mitochondria. So finally, this is so far, I think this is the only program can quantify the fission and fusion events automatically. So how do they quantify the fission and fusion event? They quantify most of them depends on volume. So if this is the mitochondria after this frame, it's divided, but it becomes 2 mitochondria. So they calculate the volume of this one and the volume of this 2. So if there's 2 plus is equal to this one, so they will say, okay, so this is the fission event. Okay, the same like fusion, the volume of these 2 after fused together is similar to, this 2 volume is similar to the volume of this one. So they will say, okay, this is a fusion event. So from here, this program gives you the, first they give you, they can automatically segment. So they can give you several parameters of mitochondria. And then they will track the mitochondria automatically, and then also track the fission-fusion events. So in this paper, they give you some example. I just take one. For example, they took the PC12 cells or non-small cell lung carcinoma or the foreskin fibroblast. So it's already known in PC12 cells the mitochondria are more like a circular form, and in the non-small cell lung carcinoma, it's like intermediate elongated, and the fibroblast is very long elongated. So they use different kinds of microscope to take the images and then compare the size. So like this is the Zeiss confocal images, this is the wide field Nikon images, and these are Olympics images. So anyway, from here they can come after use this algorithm, they know the fibroblast is indeed more elongated, and this is in the middle, and this is more circular one. They also use this one to track the mitochondria and calculate the moving speed. And then so if they give them the nocardazole to dissociate the microtubule, so the moving track of mitochondria is dissociated, so mitochondria will stop. In all different kinds of cell type, they can see the decreased speed of mitochondria. They also can check the fission-fusion event. So it's already known under the nutrition starvation, the mitochondria will fuse together, become more elongated. And if you give them some drug, for example, here is the oligomycin to inhibit the complex 5, the mitochondria become fragmented. So from different cell type, after add the oligomycin, you can see the fission event increase. And then under the nutrient starvation, the fusion event increased. So they prove that this is the automatic program can analyze mitochondrial morphology, fission-fusion, and movement. So they also use the 3D culture, and this is also pretty good for the 3D culture analysis. So finally, they compare the their mitometer to other open source method. So I have mentioned the mitograph, QVP, and the TrackMed. So notice that for 3D image, every algorithm program, they are good. They are all good. But then for 3D images, that's many limitations. So mitograph is very good. TrackMed is also very good. For like QVP, is some limitation. It's not that good for the 3D images. So like for the fission fusion event, most of the open source method is not that good. So for the mitometer is good. And then for the GUI, which is a graphic user interface, it's pretty friendly for users, especially like us. We are not that good at programming. It's pretty good for the for users. Others, they are also okay, especially actually most of them can integrate into the ImageJ. So you can use that like easier. Okay, so it's almost there. So in the following few slides, they show you how you can use this one. So this is also mitometer is written by using MATLAB language. So you can, if you are good at MATLAB, you can directly use the MATLAB version. But if you are not, you can use the ImageJ version. But for the mitometer, it's pretty easy. You just need to select whether your image is 2D or 3D. Then you enter some parameters, including the size of pixels, and then your time between frames. In the 3D, you also need to give them how many stack, how many Z stack, and then the distance between the Z stack. Then they will automatically do everything for you. So they will automatically segment it, and then this is the final image. If you are not happy with that, there are in the automatic setting, you can give them some criteria. Then you can come back to see whether you like it or not. Then after that, they will give you every parameters, including areas, volumes, and moving speed, distance, direction. So, and then after that, they can do the quick analysis for you. For example, in this example, is like they compare the mitochondrial speed in non-treat cells or treat was not non-cortisol. So you can see the speed becomes lower, and they can do the comparison for you. They will do the one-way ANOVA for you. But you can also export the raw data to do the quantification you need. So finally, if you are not happy with the automatic method, you can give them some parameters. And for example, you can define the mitochondrial area and the minima and the maxima area. So they don't find the wrong signals. Okay, so this is the final one. So because it's automatically segment, sometimes in one or some frames in your movies, they are not applied for the automatic one. So in some of them, the signal is not that good, just in some frames in your movies. So for that specific frames, you can use the troubleshoot to give them the all parameters they need to only for that frames. Then you can still do, for other frames you can do the automatically, but only for this frame you can do by manually. So I think that's it for today's method. So any questions in this one? So anyone you doing mitochondria study or related? Okay. So I hope if you, in the future, if you want to see some more mitochondria stuff, you can use this one. Okay, so that's it. Okay, thank you.